Hi everyone, my name is Delaram. I'm a third-year PhD student in Gary Beta's lab at the University of Toronto, and today I'll be talking about a project on using Varimax-PCA to understand the sources of variation within a multi-strain map of the healthy rat liver. Up until today, liver transplantation is the only treatment option for end-stage liver disease. Despite the recent advancements in post-operative care, liver transplant rejection is observed in 20 to 40% of transplant patients. This highlights the importance of studying the underlying mechanisms of this process using an appropriate model animal that can capture the liver immune microenvironment. The laboratory rat is the standard model in liver transplantation. Interestingly, liver allograft rejection is known to be strain-specific in rats. For instance, although dark agouti as the donor and Lewis as the recipient are used to model acute liver rejection, reversing this direction leads to tolerance. As an initial step towards understanding the mechanisms of rejection and tolerance in this animal model, we use single-cell RNA sequencing to understand the cellular landscape of the healthy rat liver. In collaboration with Sonia McParland and Ian McGilder labs, 6 healthy male rat liver samples, 3 from each of the dark agouti and Lewis strains, were gathered and sequenced. As we were trying to go through the standard single-cell map construction process, we faced multiple challenges. Some of the cell types in the liver tissue, such as hepatocytes, are particularly sensitive to physical stress and are easily damaged through the dissociation process. As a result of the contamination of the mRNA molecules released from the lyse cells, which are called ambient RNAs, the pathway analysis methods failed to work, and the cluster annotation and strain comparison were also inconclusive. These challenges indicate a need for an approach to separate the signal and noise within the data. Matrix factorization is a class of unsupervised techniques that can be used to discover the underlying factors that can explain the interrelationship between the observed variables. These methods decompose the gene expression matrix into the loading and score matrices. Some of the popular methods of matrix factorization are PCA, Varimax-PCA, ICA, and NMF. Here, we applied Varimax-PCA to our gene expression matrix and evaluated the resulting loadings. The standard PCA tries to identify orthogonal dimensions that capture the maximum amount of variance in your data. The Varimax-PCA applies a rotation to the PCA loading matrix with the goal of improving the interpretability of the factors. Ideally, each factor would capture a specific biological or technical signal. To interpret the resulting Varimax factors, we first identify the rotated principal components that explain cell type signals using correlation analysis. An example of a factor and a cellular identity match has been demonstrated on the right. Once a cluster and a factor have been matched, we evaluate the biological function of that cluster by performing pathway analysis on the loadings of the corresponding factor. Using this approach, we were able to fully annotate our total liver homogenic map of the healthy rat liver. And to provide a more detailed resource of rat hepatic immune cells, 2 additional immune-enriched samples were sequenced and annotated. The immune-enriched map provides a high resolution of lymphocytes and myeloid cells, and T cells, NK-like cells, B cells, and dendritic cells form individual clusters in this map. We were also interested in identifying factors that can explain strain-specific differences. To achieve this goal, we used a random forest model to predict strain labels from the factors identified per cell and discovered the factors most important to the strain label classification. The top 2 hits were Varimax factors 5 and 15. Both factors indicated near zero correlation with the QC covariates, suggesting that they have captured biological rather than technical variations. The strongest strain-specific signal is observed with Varimax-PC5. The top genes associated with this factor are hepatocyte markers, such as ApoC1 and cytochrome P450 genes, which indicates that this cluster mainly represents strain variations within the hepatocyte populations. The second strongest strain-specific signal is Varimax-PC15, which is mainly associated with myeloid population of both rat livers, as confirmed by the presence of myeloid markers among the genes with the strongest association with this factor. Pathway analysis identified higher activation of lymphocyte-mediated immune response to interferon, and allograft rejection pathways in Lewis compared to dark agouti myeloid cells. These results suggest that the baseline hepatic microenvironment in the Lewis rat is more pro-inflammatory compared to the dark agouti strain, and highlights myeloid cells as the potential key drivers of enriched inflammatory pathway activation in the Lewis rats. To functionally validate the computationally inferred strain-specific differences, our collaborator, Sai Chang in the McParland lab, performed ex vivo LPS stimulation followed by intracellular cytokine staining. We found a higher frequency of Lewis intrahepatic myeloid cells secreting TNF-alpha in response to LPS stimulation compared to dark agouti liver-resident myeloid cells. These results confirm the computational finding that the inflammatory potential of hepatic myeloid cells in Lewis is higher than dark agouti. In the next steps, we plan to extend this analysis approach and perform a joint estimation of the relevance of both observed and unmeasured factors using a scalable implementation of generalized linear mixed models. We also aim to understand how liver function is disturbed throughout the transplantation. To address this question, we have gathered samples from syngeneic, rejected, and tolerant rat livers at 2 different time points. I'm currently generating the transcriptomics map of the 3 models and aim to compare them using our established pipeline. To summarize, the Varimax-PCA analysis captured the cell identity gene expression signatures of different hepatic cell types and provided key insights into strain-specific differences in the rat animal model. Strain-specific differences were not discoverable using standard differential expression-based methods, providing a good example of the utility of factor analysis for single-cell genomics data analysis and interpretation. And with that, I would like to thank you all for listening, and please make sure to check out my poster if you'd like to learn more.